Hello, everyone. Welcome to our Crapo Award webinar for whatever the time you are in the globe, And we today we have our guest here, Dr. Xingbin Ai, who's gonna talk about her work that she did at Harvard, medical school, and she was awarded she got the Crapo Award for ATS 2025. Ridhima, do you want to explain a little bit about the award and introduce Dr. James Crapo?
Thank you, David. So it's my honor to introduce the Robert Crapo Memorial Innovation Research Methodologies Award. The award honors the lifetime achievement of Dr. Robert Crapo, an international leader in pulmonary function diagnostic research. Dr. Crapo's seminal work focused on novel approaches in standardizing lung function testing that has greatly advanced our understanding of the importance of such testing. The award honors Dr. Crapo by recognizing a mid-career researcher who is developing new techniques or approaches that show potential for enhancing our understanding of the relationship between respiratory structure and function. It is also my honor to welcome Dr. James Crapo, brother of late Dr. Robert Crapo, who would be offering introductory remarks reflecting on his brother's remarkable legacy and lasting impact in the field of respiratory medicine. So today's session will be highlighting the legacy of innovation, celebrating next generation leaders in pulmonary research, and shed light on the creative scientific thinking that propels our field forward. So thank you for being here. And now I would like to hand over to Dr. James Crapo.
Oh, thank you. Let me share my screen. So basically, it's a real honor to be asked to say something about my brother and what he contributed to the field. This is a picture of him, a recent picture before he passed away. I would tell you that my brother and I both went to the University of Rochester. He's 2 years older than I am. And he attended the University of Rochester with me in the 1960s. And then he went to the Navy for 2 years during the end of the Vietnam War. And what I want to tell you is that this was the beginning, really the very beginning of pulmonary medicine. The pulmonary specialty started at a few medical centers in the late 1950s, 1960s, but it was not universal. And at the University of Rochester, where we trained in the 1960s, there was no pulmonary medicine program. We had one pulmonologist from the community come in once a week to our internal medicine rounds. And there was also no ICU. So after Bob, he actually chose after the Navy to go to the University of Utah, which actually had a really outstanding pulmonary program under Till Renzetti. And as he finished that, Bob went to on the faculty in the early 70s to the Intermountain Health Hospital. And he was placed in charge of the pulmonary function unit, and critical care was just starting. I don't know, people probably don't realize that, but when I went to Duke, and in the early 70s, the first ICU was set up by taking out the wall between a couple of patient rooms and made room for 6 patients to be on ventilators, which they were just developing. So I'm speaking at an earlier stage of development. But as Bob went into the pulmonary function lab, he was excited by the opportunities as we were measuring all kinds of things, but no one knew what they meant. There was no data, no standards. And he spent years at the very beginning testing lots of people to find out what normal was and what was abnormal and how it related. And so he published, he led the publication of this book in the early 80s from the Intermountain Thoracic Society that set standards for pulmonary function tests and set the normal ranges for virtually every part of it. That became a standard book that was used worldwide for several decades. And it was actually adopted by every single manufacturer of pulmonary function equipment and incorporated into their software so that their outputs would use the Crapo standards, so it's called, during that period. And I just tell you a little anecdote. I would go to give a lecture someplace on the East Coast, and inevitably someone would come tell me their pulmonary function machine was named after me because at the bottom of the king was the word Crapo to tell people whose standards they were incorporating to their reading of their tests. But it brought a level of rigor and standard to the field that was unknown at the time. At the time, we really needed it, and he was incredibly rigorous about how they did the test and how they interpreted it. And then he became the chair of the committee for both the ATS and ERS to review standards for pulmonary function testing. I'd say he also did the same thing in the ICU, which was, the intensive care medicine didn't become a separate specialty until the 1980s. And the ICU set up that he and Alan Morris set up an ICU in Utah that was unbelievably rigorous. And I remember being totally amazed at what he was doing and asking him how he could so easily take care of the really critically ill patients that they brought in there. And he said, it's very easy. What you do is you put a line or a device into everything you can, and you measure it, and then you normalize it. I liked his life. He began with rigorous data, and he laid a foundation for the whole field of pulmonary medicine to use data rigorously to accomplish their goals, and had a profound effect on the field at every stage throughout the world. I just go on to say he also loved Utah. He loved outdoors, and my favorite picture of him is one I'll show you right here. My favorite memory is actually floating down the Green River in eastern Utah, looking for an eddy where we could find a fish like this. But he put the same rigor into tying flies that he put into measuring pulmonary function tests. So he lived his life that way and was an inspiration to me, and I think laid a foundation for our field. So it's a real honor for a family and for me to be here and for the family to have this special lecture to recognize some of the rigor of the medicine. And today we're going to learn about respiratory infection in infants. So I'll turn it back and say thank you. It's a wonderful honor to tell you about Bob. I need to stop.
Yes, Dr. James Crapo, thank you very much for this wonderful introduction, And in fact, back at time 1997, when I started pulmonary medicine, I remember seeing on spirometry, the Crapo, as a standard, for the normalization. So let me introduce Dr. Xingbin Ai, which we overlap a little bit the time, we spend in the Boston. So the Dr. Xingbin Ai, she started her career, by attending her undergrad bachelor in genetics at the Fudan University, which we have a few pulmonary scientists that graduated from the same school, And then she came to United States, and then she completed her PhD in May of 2000 in neurosciences at the Case Western Reserve University. And then later on, she moved to the Pennsylvania for attending her postdoctoral training until August of 2005 in cell and developmental biology. She's right now a faculty member at the Harvard Medical School. She's based at Mass General Hospital, where she's a director of the basic research in newborn medicine. And for many, many years, she was she have a fundamental research, and key research into understanding to understanding neurons and the lung function. And after she moved to the Mass General, and before that, she was at the Brigham and Women's Hospital at the Pulmonary Critical Care Medicine. Basically, she developed many innovative methods to understand the lung biology, including generating cells from the bronchoalveolar lavage as well as a PCLS. Okay, I will give, I will let Dr. Xingbin Ai, on this seminar. Thank you very much.
All right, so I have to say it is an incredible honor to be the speaker of the ROC Memorial Lecture in 2025 in memory of Dr. Crapo's excellence and innovation in lung research. I also want to thank American Thoracic Society and all the colleagues and friends who have been supportive of my career, and also the people who have been working behind the scene to make this webinar possible. So today I'm going to talk about some of our ongoing effort in the lab to model respiratory infection in infants. So I guess I want to give you this cartoon to highlight a key point. The point is young children are not just little adults. As we all know, the human lung continues to develop after birth, and then the lung is also constantly challenged by the environmental insults. This include, allergens, pathogens, pollutants. A lot of study from my lab, as well as others, have highlighted the profound impact of these early life exposures on the lung developmental program that ultimately leads to changes in long-term lung function and the structure and the disease susceptibility later on. So in my own group, we have shown not only airway epithelial cells, but also smooth muscle cells and the nervous system. They respond very, very differently to environmental allergen exposure, for example. Compare the neonatal lung versus the adult lung. So I guess all these findings highlight the importance of establishing age-related as well as physiological relevant models to study pathogenesis of diseases affecting infants and young children, the most vulnerable population in the human race. So one of the diseases that's dearest to my heart as a fundamental biologist at the MGH in the surroundings of newborn medicine is respiratory viral infection, specifically respiratory syncytial viral infection. For some of you who may not be familiar with this virus, I want to make a very brief introduction about RSV. So RSV is a single-stranded RNA virus. The primary target of RSV, let me get my pointer. So the primary target of RSV is ciliary cells in the human bronchial epithelium. RSV is the primary cause of bronchiolitis and hospitalization in infants. RSV bronchiolitis is characterized by epithelial cytopathy, mostly apoptotic cell death, and refractory airway hyperconstriction, leading to difficulties of breathing in the affected young patients. RSV is a big problem worldwide. In the United States alone, more than 58,000 hospitalizations occur each year for young children younger than 5 years of age due to this virus infection. And worldwide, there are about 100,000 deaths worldwide for this very small patient population. So the reason why this disease is devastating is currently there's no effective treatment of active infection. We have developed prophylactic approaches as well as vaccination during the late pregnancy that have been shown to reduce hospitalization and then reduce the frequency of infection. However, the protection is not 100%. For RSV, active RSV infection in infants and young children, there's no effective treatment. The current treatment remains supportive care. So why RSV has been such a big problem for neonates? Partly because RSV has a very, very narrow viral tropism. So I just want to give you one example. This is a paper published in 2013 by Dr. Pickle's lab. What they have shown is comparing bronchial epithelium in vitro from mouse, hamster, non-human primate, and humans. They compared 3 different viruses. They are all RNA viruses. How these viruses infect these epithelial cells from different species. As you can see, the mouse parainfluenza virus can infect the different species very similarly. However, the human RSV predominantly or almost exclusively infect human bronchial epithelial cells. The infection in non-human primate bronchial epithelial cells is very, very limited. And then this virus just doesn't infect the rodent epithelial cells. So these narrow viral tropism greatly limit the animal models to model the infant infection by this devastating virus. So there are many other reasons, of course. I want to highlight another major reason, in my view, why the rodent model may not be fully recapitulate the disease pathogenesis of this devastating infection is due to the differences in the structure and the cellular components. Here is a cartoon I took from a review published in 2014 by Dr. Bridget Hogan. What you can appreciate here is look at the human small airway. This is the small airway that are infected and then can cause severe damage, and also the airway flow blockade. So these epithelial cells in human small airway represent a pseudostratified layer with basal cells at the bottom of this layer, and I will come back to this cell type later. However, if you look at the mouse lungs, the only structure that looks similar to the human bronchial epithelial cells in the small airway actually are the tracheal epithelium. If you look at the intrapulmonary airway of the mouse model, the epithelium is very thin, is predominantly small, and it's almost looking like towards the terminal bronchial region of the human lung. So because of the structure, cellular differences between human lung and mouse lungs, you would imagine the cellular response could be very different on top of a very narrow tropism of RSV limited to human epithelium. This really creates a lot of technical difficulties of studying this devastating disease. So before I move into our study, I would like to highlight a very interesting fact, which is, although RSV can cause severe infection in newborns and infants or children younger than 5 years of age, RSV infection in healthy adults younger than 65 years of age barely cause any significant symptoms. So why age significantly affect the infant? Why age significantly regulates the severity of RSV infection is not very fully understood. There are many studies comparing the immune system that differs from neonates versus adults. The immune system likely contributes to the susceptibility to severe RSV infection in infants. However, because RSV predominantly impact on the epithelial cells, we also believe that epithelial cells between the age groups may also contribute to disease severity. So to do that, we need to create relevant and age-appropriate models to model RSV infection in infants. So this comes to the talk. Hopefully in the next 30 minutes, I will present to you the new models that we have created, both in vitro and in vivo and ex vivo, that really recapitulate the disease pathophysiology observed in a clinical setting in infants. And then this opens really the opportunity to really study age-related susceptibility, as well as identifying potential therapeutic targets. So the first model I'm going to introduce is an in vitro bronchial epithelial cell model in air-liquid interface culture of human basal stem cells. And then the second model I'm going to introduce is an ex vivo model that has to do with precision cut lung slices generated from infant donor lungs. So as I mentioned earlier, there are many differences in terms of the cellular composition between human lungs and mouse lungs. So here, one of the major one of the major differences in the cell type in a human lung is the basal stem cells. The basal stem cells are this group of cells. They reside at the bottom of this pseudostratified bronchial epithelium. You can recognize these cells by their unique expression of TP63, which is one of the transcription factors selectively expressed by basal cells, as well as other markers, for example, keratin 5. In the human lung, basal stem cells reside throughout the respiratory epithelium, the airway epithelium, and then these are one of the major stem cells that can generate the entire differentiated bronchial epithelial cells. The traditional way of isolating basal stem cells rely on either lung biopsy samples or bronchial lavage fluid. These two procedures are extremely invasive and are often contraindicated in infants who suffer respiratory diseases. So just give you some examples of how bronchial basal stem cells can be utilized to model the human bronchial airway epithelium. You will understand why it is very important to isolate, to have a method that can readily isolate the bronchial stem cells from infants or the age-relevant groups. So the basal stem cells can be cultured, as I said, traditionally isolated from lung biopsy samples and the bronchial lavage fluid. Can be cultured in expansion medium, and after that, you can seeded cells in an insert on the top of this insert is called air-liquid interface here. Once the cells reach confluency, you can remove the medium from the apical chamber to expose the basal stem cells to the air. That triggers the differentiation of the basal stem cells into multiple major epithelial cell types. Here is a section of this day 21 AI culture of basal stem cells. You can appreciate the structure and the cellular types in this AI culture reproduces what we found in the human bronchial epithelium in this small airway. For example, the basal stem cells, these are remaining non-differentiated basal stem cells. You can find them by antibody staining of P63. And then they reside at the base of the pseudostratified epithelium, same as you found in the in vivo. You can also identify differentiated secretory cells identified by the expression of CC10, goblet cells by the expression of MARC5AC, and most importantly, ciliary cells identified by their expression of the ciliary cell marker, acetylate tuberin, is labeled the mobile cilia on the apical side of this bronchial epithelium. So this model truly creates a wonderful system to study human bronchial epithelial cell responses to various diseases and also various allergens and pathogens. However, as I pointed out, the traditional effort of isolating basal stem cells relies on biopsy or bronchial lavage procedures. These procedures are invasive and often contraindicated in newborns and infants. So before I joined MGH, Dr. Paul Lalou, who is a neonatologist at the MGH, he discovered already the trachea aspirate actually contains very rare basal stem cells. So the trachea aspirate is considered medical waste. The trachea aspirate has to be removed before the intubation tube can be put in space. So traditionally, clinically, nurses using a suction remove the trachea aspirate and then toss it away. But rather than throw it away, we decided to collect all these trachea aspirate. And then we found the trachea aspirate actually contains a variety of human lung cells. So you could see the immune cells. In this particular image, we show the presence of a large number of phagocytes, likely alveolar macrophages, because they can engulf the fluorescently labeled beads. And then we also know the trachea aspirate contains a small amount of mesenchymal stroma cells that can be amplified and used to study how neonatal diseases such as infection modulate the fibrosis process in the lung. For today's talk, I'm going to focus on this very rare cell population in trachea aspirate, which is basal stem cells. As you can see, this is basal stem cells is APCAM positive, and then these cells are stained positive for the marker of basal cells P63. So by working together with Paul Lalou's lab, both of us are working very diligently over the past 5 years. We have built a bed to the bench pipeline where we can collect the endotracheal tube suctioning, move the suctioning to the lab, isolate the different cells, and then usually within a week or two, we can get significant expansion of the basal stem cells in the growth medium. We usually amplify the basal stem cells a little bit longer, so after 6 weeks, we can easily get millions of cells for additional experiments. So some of the cells we can store in the liquid nitrogen, and then we can use other cells to perform all kinds of experiments, including the talk today, the modeling an RSV infection in newborns. As I said, because the trachea aspirate is considered medical waste and is a standard procedure in patient care, this procedure opens to new ways of isolating basal cells from patients that using traditional methods is not possible. So using the basal stem cells isolated from trachea aspirate, we're actively modeling not only the infection, but also study how prematurely infection in neonates, as well as congenital diseases, how they affect the epithelial differentiation. So for today's talk, I'm going to focus on basal stem cells harvested from trachea aspirate of patients who suffer from the cardiogenic and neurogenic conditions, and otherwise they have normal lungs. So in other words, these basal cells are collected from neonates who are normal and the young infants who are normal. They don't have lung diseases. So here just shows you again what the collection and the derivation looks like. This is the suction cup, contains usually is less than 1 ml of trichic acid from newborns. And then what we can find the very few basal stem cells in this culture medium. After purifying with the APCAM-Bs, we can put this epithelium enriched portion in the culture medium, and usually within a week, we start to see the expansion of the basal cell clones. We know these are the basal cells because they stand for P63 in 100% of these cells. So when I showed this data, all the people I talked to asked me a very important question. The question is, how do you know where these cells are coming from? Are you sure these are the meaningful bronchial stem cells? So usually the answer for me is we actually don't know where these cells coming from. We do believe these cells likely get suctioned out because either the disease caused the epithelial damage or the procedure of putting the tube in causes some damage to the epithelium. That's the reason why we have very small number of basal cells. But to effectively address the question, what's the identity of the trichic acid-derived basal cells? What we decided to do is to compare the trichic acid-derived basal cells to nasopharyngeal cells. These are the cells isolated from newborns using the nasal swabs, and the cells that we can isolate from a very few number of infant lungs. We can isolate small airways. These are the airways less than 1 millimeter in diameter, as well as extremely small bronchial area. And then using traditional methods, we can isolate the basal cells from small airway versus bronchial region from infants. So we performed many different studies. I just want to show you the transcriptomic profiling study that will probably illustrate the points very easy. So the first thing is we know the trichic acid-derived basal cells are not nasopharyngeal basal cells. This is the transcriptomic profiling. You can see the trichic acid-derived basal cells are further away from the nasopharyngeal basal cells. If you look at the genes that are selectively expressed in the trichic acid basal cells, you would identify NKX2.1, which is a lower respiratory epithelium marker that's missing in the nasopharyngeal basal cells. In addition to that, we know the trichic acid-derived basal cells transcriptomically overlays with the bronchial basal cells, but not from the bronchiolar. So in another word, based on the transcriptomic profiling, as well as the differentiation study that I did not present today, we know the trichic acid-derived basal cells are very similar to bronchial basal cells that can be utilized to generate bronchial epithelium for disease modeling. So because we have these cells, we are very excited to test whether age has any impact on epithelial cell infection by ISV. So to do that, we have to answer first a few questions. The first question is, does the age affect the abundance of the ciliated cells? So to test that, we did a number of studies, including using the neonatal trichic acid-derived basal cells compared to adult trichic acid-derived basal cells. We also look at the abundance of the ciliated cells using histology in vivo. So the conclusion is very clear. The age doesn't really affect the differentiation capacity of the basal stem cells. As you can see, the ciliated cells, which are the primary target of ISV, are labeled by IFX3, is a transcription factor selectively expressed by ciliated cells, as well as acidated tubulin. This is the marker for the cilia. And then again, this is a CC10 for secretory cells, MARK5AC for garbage cells, and the TP63 for the basal cells. Based on the quantification of histology, we know the proportion of ciliated cells between neonates and adults are very similar. So the next question is, does ISV adhere to the neonatal bronchial epithelium or adult bronchial epithelium differently? So to do that, what we did is we incubated RSV on the apical side of this AI culture. We waited for 1 hour at 4 degrees. So this is the condition where the adhering can occur, but the internalization of viruses cannot occur because the internalization requires a biological active process that requires cells be incubated at 37 degrees. The answer is the virus seem to adhere to the apical side of the neonatal bronchial epithelium and adult bronchial epithelium very similarly. So how about replication? So for the replication, what we did is incubate the virus for 1 hour on the apical side, washed away any virus that are not adhering to the apical side, and then waited for 6 hours at the 37 degree. This is a time that allows the primary replication of the RSV. Again, we don't see any age-dependent differences. In addition to that, we also followed this culture over time. This is the staining for the RSV using F protein antibody. And you could recognize, very similar to what we found in the early phases, at day 1 post-infection, we don't see significant virus spread between the 2 age groups. The number of infected cells is very, very similar between age groups. So the age-related infection, the age doesn't seem to impact on the initial RSV infection during the early phases of the infection process. However, just by waiting for 1 more day, what we found is very, very different. Here is a typical standard represented images showing you RSV labeled by antibody in green, and also the other marker, 01, identifies the tight junction between the differentiated epithelial cells. Tight junction formation is a very important feature of the barrier function of the bronchial epithelium. So as you can see at day 2 post-infection, the neonatal culture shows significant RSV spread, evidenced by about 30% of cells now have RSV intracellularly. Compared to the adults, the number and the percentage of infected cells seem to remain relatively low and not that much different from day 1 infection. And at day 4, the significant virus spread continues in the neonatal culture, but the adults still maintain a relatively low percentage of infected cells. So this is the first evidence that RSV infection, the severity of RSV infection can be regulated at the bronchial epithelium layer by age. Importantly, amongst area where RSV spread can be detected, we also see significant damage in the 01 labeling, indicating the barrier damage that can be also measured by the reduction of the transepithelial electrical resistance. So this is another measurement of the epithelial resistance. So this is just a quantification of the percentage of RSV infected cells. So we know the RSV can kill the cells. So we decided to look at whether the cells infected with RSV in the neonatal epithelium are undergoing cell death. We tested different cell death markers. We found apoptotic death is the prevalent way of cell death in the neonatal ciliary cells infected with RSV. So here the cleaved caspase 3 is a marker of apoptosis. It's stained in red using antibody. And you can see increasing number of apoptotic cell deaths in the neonatal culture starting from day 2. In the adult bronchial epithelium culture, we actually do not find significant cell deaths with time, even after 4 days. In addition to the staining, we also confirmed the results of increased apoptosis in neonatal culture with Western blot. Here you can see with RSV infection on day 2, we have a significant increase in the level of cleaved caspase 3 compared to lack of change in the adult culture. So overall, you can see the RSV infection caused significant apoptosis, virus spread in the neonatal bronchial epithelium, but these events do not happen in the adults. So we're very, very excited by these findings because based on a few histological studies of fatal cases of infant RSV disease, we know the RSV can cause apoptotic cell death predominantly in the bronchial epithelium cells. Here is a staining presented by the paper published in 2007. The caspase, cleaved caspase staining in the fatal cases of RSV in the lung is predominantly located to the epithelial cells claimed by the author. Very interestingly, in the fatal cases of influenza infection in infants, the authors also detected cleaved caspase in the lung sections. However, all these caspase 3 stained positive cells actually are immune cells. So again, this really highlights a very, very important virus-host cell interaction and the virus-specific mechanisms that trigger the cell deaths. And for RSV, the apoptotic cell death is one of the major reasons for all the pathogenesis happening in the infants. So in addition to widespread virus spread, widespread RSV, and the cell death. We also look at the mucus hyperplasia. The mucus is stained in green using an antibody against MARC5AC. So as you can see here, we can find a significant increase in MARC5AC production after 48 hours of post-infection. And then more importantly, the mucus layer was so pronounced in the neonatal culture following RSV infection at day 4. But in the adults, the number of garbage cells actually remains relatively unchanged. This finding is also very important because in the fatal cases of infant RSV disease, mucus hyperplasia wrapped around these dead cell debris are one of the key features of pathology. So this is basically showing you the mucus, the cells inside mostly are dying epithelial cells. All right, so basically this in vitro model recapitulates the epithelial pathology as we found in vivo in severe RSV infection in infants. So utilizing this in vitro model, very simplified, we started to understand more and more about how age determines the severity of epithelial infection. So the paper published last year identified an age-dependent STAT3 activation response to RSV that determines the severity of RSV infection. So in contrast to adult ciliated cells that can elicit a strong STAT3 activation process to turn on anti-apoptotic gene to keep the cell survival, the neonate somehow is impaired. And then the apoptotic cells in following RSV infection is utilized by the RSV to spread the virus. So this really brings out a very important epithelial mechanism that controls the severity of RSV infection. So we also are very, very interested in additional viruses, as I highlighted, comparing influenza and RSV. The viruses clearly utilize different mechanisms of triggering epithelial cell death, as well as virus spread. And we are very, very intrigued by SARS-CoV-2 infection, because as you probably all know, the SARS-CoV-2 infection actually have the opposite age-related severity. The young children actually are somehow protected from getting severe disease, but the adults are more prone to develop severe infection. So using our model, we know the SARS-CoV-2 does not appear to show age-dependent infection in the bronchial epithelial cells using our culture system. And very interestingly, SARS-CoV-2 also did not induce STAT3 activation. The STAT3 activation actually was found in a bystander cell, likely in response to the cytokines. So here, I just wanted to give you this example. Our system can be potentially used to study other type of viruses, and then to study how virus and how virus-specific interaction with host cells may regulate the pathogenesis of lung damage. All right, I guess I want to give a very quick intern summary of this in vitro model. I think I want to convince you we have developed a very robust method using tracheal aspirate to derive basal cells. This new approach potentially facilitates greatly the basal cell isolation without any invasive procedures. And then this procedure opens new ways to study epithelial defects for neonatal as well as infant diseases that traditional isolation methods cannot be possible. So I also showed you in this part of my talk, age actually regulates susceptibility to severe RSV infection in the human bronchial epithelial barrier. And then lastly is the neonatal IR culture model I showed you can be potentially used to study a variety of viral interaction. And for the RSV, we can use that also potentially as a screen for drugs and testing therapeutic agents. But after all these talks and all these findings, we're still not happy with the epithelial model. The reason why is in the infant, epithelial cell death is not the entire pathophysiology that has been observed. As I mentioned earlier, the refractory airway constriction is another key feature of the severe RSV infection in infants. So how can we model airway contractility phenotype in this devastating condition? So we decided to use precision cut lung slices from neonatal and infant mouse to see whether we can recapitulate some of the airway physiologic changes observed in vivo. So I think if if you still remember, Dr. Ramakrishnan last year presented this webinar. In his presentation, he showed elegantly using adult human lung sizes to model changes in lung mechanics in different chronic adult diseases. He also presented his work using normal adult human lung sizes treated with different type of signals, including cytokines and fibrotic signals, to model fibrosis. So here, my lab has worked long-term. Rama is my long-term collaborator. Both labs have worked together over the last decade. We co-developed a cryopreservation method to preserve live and functional lung sizes prepared from mouse lung and as well as the human lung, so that the experiment can plan as you want. So just to give you some brief idea how we prepare this, in case you did not remember what Rama has presented last year. So what we have to do is we have to infuse 37-degree low melting agarose, about 2%, into the lung so the lung can be inflated. And then after solidification, the agarose also provides the support for slicing. Using a lung slicer, we usually collect lung slices that is about 250 micron in thickness. The lung slice is alive. You can find the cilia beating under the microscope. But more importantly, it kept all the it keeps all the structure and the cellular components you can find in the lung. So this included the airway epithelium. We also know there are a layer of fibroblast cells and smooth muscle cells, as well as many endogenous immune cells, as well as the alveolar space. So using these lung sizes, we decided to do much more elegant work to follow RSV infection using live imaging. So in this particular study, we're using an RSV virus tagged with GFP, so you can actually follow the infected cells over time. So this is this is the same airway. This is a low mag picture just to show you very few epithelial cells are infected with RSV. But if you just wait one day, there's 2 days post-infection, we see widespread RSV infection in the neonatal bronchial epithelium in the infected lung slices. We know these cells are ciliary cells because you can stain the cells, fix the slides and stain for acidated tubulin, which is a marker for ciliary cells. And you can see these are RSV infected cells. They all they lost the cilia. We have other markers to confirm these are truly ciliary cells. And a very strikingly, just consistent with what we found in the in vitro bronchial epithelium cell model, the adult lung slices also do not get a very low infection, and then the RSV does not spread in the adult lung slices. So basically, that reproduces what we found. The RSV can spread significantly in the neonatal and infant bronchial epithelium, but not so much in the adults. So we can do more elegant work to actually follow individual cells over time and then start to appreciate very, very unique processes called extrusion of the epithelial cells. So here, just an example of a 2-day post-infection of infant lung slices infected with RSV. In here, you have 4 cells infected with RSV. We can identify 3 cells express significant amount of active caspase. These cells all started to round up. The cell shape is very different from normal cuboidal shaped epithelial cells. They are detached from the attachment to the basement membrane. They are basically being squeezed out or shredded off from this layer. And then we can use different compounds to test the therapeutic benefits. For example, in this particular experiment, we tested an inhibitor of the caspase-3 to reduce the RSV spread. And then you can see here, the slices treated with this caspase inhibitor do not have widespread RSV infection. And even in the cells that are infected, because they're not undergoing apoptosis, the cells look in much better shape in terms of the cell shape, as well as apical cilia formation. So here, just giving you another example. This is a much zoom in the picture, showing you 2 ciliary cells at the different stages of infection in the neonates. So here is 1 cell that's almost completely squeezed out from this epithelial layer. Why we say that is you can identify the nuclei actually translocated from normally the basal side of the cell to the apical side of the cell. In addition, the cell shape become a water drop, inverted water drop. This cell probably is at a relatively earlier phase of infection because we can still detect some cilia. These are ciliary tubulin, although the signal is much reduced. However, these cells still have the nuclei at the base of the cell body. And then you can follow what is some of the cells in the treated condition. And again, this is long slices treated with the caspase inhibitor. You would appreciate the cell morphology is maintained properly, and also the cilia loss is also significantly reduced. So all in all, I felt this is a very, very unique model, and then not only allow you to study truly pathogenic changes happening in the epithelial cells within a normal lung structure, it can also provide a very, very important tool to screen for drugs. So ultimately, as I said, epithelial infection is not ultimate goal. Epithelial infection is the primary trigger, but how about other disease conditions? So the lung slices provides a perfect model to look at how the other events unfold following epithelial infection. So one of the major events that we know is the refractory airway hyperconstriction in infants, rendering difficulties of breathing. So in this particular experiment, these are infant lung slices. This is a mock control slice, and you can see there's an airway. And then the airway contracts in response to histamine. And then if you relax this muscle using beta agonist, you can see the airway movement start to open up over time. However, compared to the mock control, we found the ISV infected infant lung slices, although limited just to the bronchial epithelium, can cause profound heightened airway contractility in lung slices. And then most importantly, this contraction is resistant to relaxation in response to beta agonist. So this is exciting. It's really, this finding is very exciting because traditionally, people believe the refractory airway relaxation is because drug cannot be delivered to the site. Here, using the lung slices, we actually indicate that these results actually indicate the airway smooth muscle cells must have changed following an epithelial infection, because in the lung slices, the accessibility to beta agonist is not a limitation. So that we are very, very excited by this finding. And currently, we are actively studying how the airway smooth muscle cells become dysregulated in their relaxation response. So just to better show you what we found in the ISV-infected infant lung slices, in addition to heightened contractility, this is ISV-infected lung slices, the luminal shrinking is significantly enhanced. More importantly, the contracted airway can no longer be relaxed by beta agonist. In contrast, the mock control can be relaxed very readily. Here is just a quantification. Each color represents one donor in this experiment. So as I said, the lung slices can not only reproduce the epithelial phenotype, also reproduces the airway contractility phenotype. And then we are actively pursuing potential therapeutic testing. And this is just one example. As a proof of principle, we show this RSV-induced hypercontractility can be relaxed if you just activate cyclo-MP directly using boscaline. So this really indicate the mechanism that prevents airway from relaxation following epithelial infection of ISV likely happens at the receptor level, not at the intracellular signaling level. And then, so our finding also brings out a very important communication between epithelial cells and the smooth muscle cells that ultimately cause deregulated airway contraction. So we can start to use our model system. Now we are using co-culture system. Here you collect the secretion from the epithelium following ISV infection. And then we're using this secretion to treat infant precision cut lung slices. And after 48 hours, we found identical phenotype as the ISV infection directly in the infant lung slices. So we are currently pursuing what exactly is produced by the neonatal AI epithelial cells following RSV infection, and what is the receptor in the smooth muscle cells that respond to this disease-induced signal and ultimately leads to the relaxation defects. So you can see here, these findings indicate the first one is airway smooth muscle cells become dysfunctional. Second one is this dysfunctional is due to aberrant epithelial communication. And the third one is if we can block this communication or block the airway epithelium infectivity, then we probably will be in a much better shape in treating the active ISV infection. So I guess I will summarize the talk. So I hope convince you we have an in vitro model generated using basal cells derived using tracheal aspirate, which is a very novel method, and it greatly facilitate the procedure to isolate BSC cells from patients of all different age groups, as long as they have an intubation tube. And then I also hopefully convince you the precision cut infant lung sizes provide an excellent ex vivo model to evaluate airway physiology and the communications between multiple cell compartments. And then these two models can be expanded to test a variety of pathogens and also to identify potential therapeutics. So I guess with this, I will conclude my presentation. And I want to thank the people who is currently in the lab, especially I want to thank especially Paula Liu, who is a physician scientist and has been working closely with my group to establish the tracheal aspirate basal cell model and use this model to study bronchial defects in different diseases. The majority of the work related to airway smooth muscle physiology is performed by Dr. Xingbin Ai. For many of you probably know her. She's an excellent lung physiologist, and then she's also a physician scientist. She's she's moving on his career ladder to become independent. And then related to epithelium in vitro model of ISV infection, the work is predominantly performed by a very talented postdoc, Chi. And then he has been instrumental in building up the model, especially the ISV infection model, to allow these studies. And I want to thank my collaborators, Dr. Krishna. He gave the webinar last year, and then he is my long-term collaborator. He really guided us in terms of understanding lung physiology, lung mechanics, and then the application of slice using lung slices. Dr. Mel is a collaborator at MGH. She helped us troubleshooting many basal cell culture difficulties at the beginning of establishing tracheal aspirate-derived protocol. I also want to thank Rachel and Elka at the BU. These are the true virologists. Rachel is an ISV virologist. She guides us through the entire process of how the ISV infects, how they replicate. And Elka's lab has been devoting unselfishly their time to help us setting up infection with SARS-CoV-2 and other deadly viruses. And most importantly, I want to thank patients, families, and the donors for their willingness to share their samples and cells to make this study possible. Thank the NIH for funding. And then I thank you all for listening. Thank you very much.
So thank you very much for giving this beautiful presentation. Dr. Ridhima and myself, we are moderating this session, and we want our participants to ask a few questions. I believe we have like 6 minutes for Q&A. So you can raise your hand, so then Daniel can open your microphone, or you can ask your question into the Q&A chat. Okay, I will start, with one question that on this beautiful talk. And Dr. Ai, have you ever considered looking, for example, airways from elder, compared? So now you know the story about the neonatals and the adults. I was wondering about elders and the susceptibility for the lung infection. And also, I believe your model can be very useful for development of drugs especially for those different viruses, like antivirus or maybe modulators of the response in the lung.
Yeah. So I think how about I answer the first question first, which has to do with infection of elderlies. Because if you look at the distribution of patients who are susceptible to severe ISV infection, there's the two spectrums, right? The spectrum covers the early first 6 months and 1 year, and then the other high frequency is occurring in patients older than 65 years of age.
Yes.
We have not expanded too much into the aged spectrum. To be honest with you, we have derived older patients using the same approach, because you could imagine the older patients who needs intubation because of other conditions, heart defects or other things. The difficulties there is many of these older patients have comorbidities. With age, there is usually they have other conditions that can potentially profoundly impact on the bronchial epithelium responses. So we actually see quite a bit of variation in the older group. That doesn't mean we should not continue, but that just means we probably have to expand this population to really understand to what extent or to what fraction of the patients the epithelium is one of the site to dictates the severe infection. You could imagine there are other mechanisms like immune cell components, which we don't have that.
Yes.
Yeah, so I think to answer your question, the short answer is we don't know yet, but I think it needs just probably more work than the neonatal model we are creating. The beauty of neonatal model is everybody is innocent. There's no prior infection at all.
Yes.
So I think that creates a very, very clean background to impose this virus infection because the cells have never seen any infection before. This is a very different situation in the adults. But I think this is definitely something we are working on. We probably just need much bigger resources to really tease apart how many of the patients the disease susceptibility is mediated at epithelium level, and then if they are, what is the mechanism? And then going back to your second question is whether we have considered using the models we built to screen for drugs. Yeah, definitely. We are hoping, if funded, to carry out a small molecule screen using initially the in vitro model to find a few hits. And then these hits can potentially move on to the long slice model. So basically, we have built a pipeline for the screen, starting with the in vitro model, and then we can continue on with the ex vivo model.
Beautiful. Is any question from the participants? So Dr. Ridhima, Dr. James, do you have a questions?
I can ask a couple of questions. Thank you, Dr. Ai, for a beautiful talk. First of all, my question is, did you look at in your first part of your talk, you talk about that it could be associated with cell death, the infection in neonates. Could it be any other caspases related, or could it activate inflammasomes or something like that?
Yeah, good question. So Ridhima, so we have looked at other modes of cell death. If you look at day 2, the majority of infected cells started to undergo apoptosis. But if you look at a little bit later, like day 3 or day 4, we also started to find other modes of cell death, for example, necroptosis. So one possibility is virus may trigger apoptosis as a primary direct way of causing cell dying. And then the necroptosis could be caused by inflammatory signals secreted by already infected cells. For example, interference. Interference is known to cause cell death. So to answer your question, we think other modes of cell death are likely involved, but we believe the primary way of triggering cell death by the virus is likely the apoptosis.
Okay. So my second question from your second part of your talk is, so you do see when you take the supernatant from your infected ALI cultures and infect your PCLS. So do you think it's the cytokine storm that's causing airway hyperresponsiveness?
Yeah. Great question. So we have one paper currently resubmitted under review by Blue Journal. We identified one key cytokine, which is CXCL11. So these cytokine signal very differently from CXCL10, for example. It is signaled predominantly through a scavenger receptor that can interact with the beta agonist receptor to desensitize the receptor. So you're 100% correct. The signal is from epithelial cells following infection is one of the cytokines that are responsible, at least partially responsible for the lack of beta-2 agonist response.
Okay, great. Thank you. I will hand over to James if he has any questions.
So, James, you don't have any questions?
No.
Thank you. Yeah.
I believe I don't see any questions for other participants. Okay, great. Thank you very much. Ridhima, do you want to do the final remarks? Or?
So thank you for being here, everyone. And I would extend my thanks to our speaker and James Crapo for this session. And we are grateful for everyone being here. Thank you for attending our webinar today.